All right, girls, I know we started an overview of mitosis and meiosis in class, but I'm going to go through the specifics of what's happening in each stage of both mitosis and meiosis today. This video will be just for mitosis. Most of the cells in our body, besides our gametes, are going to go through mitosis. The purpose of this is to replace all of our old cells with new ones and to create from one mother cell to identical daughter cells. And we will get a full set of chromosomes in each new daughter cell. And remember that these are going to be completely identical, the two cells that we get. All right, so our first phase, we're going to start off with our mother cell here. We are going to go through interphase. So give me one moment while I draw this. All right, girls, so here we are in interphase, and remember that this is where a cell is going to spend about 90% of its life, is going to be just in preparation for mitosis. So a couple things I wanted to point out about the structure of this cell. We have our centrosomes, which are going to help in the production of our spindle fibers. Our spindle fibers pull apart those sister chromatids in mitosis. And then we have our nucleus, which is very apparent still in interphase, with the DNA in chromatin form, which is that unraveled spaghetti-looking DNA. Now in interphase we have our three stages. We're starting off in G1, which is our first gap phase. In our first gap phase, we have just general cell growth. It's going to be the main thing that you want to know about this. S is really important. This is where our DNA synthesis occurs. All of our chromatin here is going to be replicated in preparation for mitosis. Finally, we have our G2 phase. Once again, another gap phase in preparation for the mitotic phase. We're now moving on to prophase, which is the first step of mitosis. Remember that interphase is technically not part of the mitotic phase, just prophase through cytokinesis. Cytokinesis is part of the mitotic phase, however, is not part of mitosis. You saw that in the cell cycle pie chart that I showed you guys in class. So in prophase, what we have here, our centrosomes are starting to move to opposite poles. We're going to see them continue to move down this way. Now, also our nuclear envelope, our nuclear membrane, is starting to disappear. It's not quite as apparent anymore. The last important thing to note is that our chromosomes have now become condensed. So instead of being in chromatin form that they were before, where they were kind of that spaghetti-like shape, now they're actually forming into these X's. Remember, each side of our chromosome here, this is one chromosome, is made up of two sister chromatids. So here's one sister chromatid, and here is the second. Those two sister chromatids are exact copies of each other. They're completely identical. So then these are eventually going to be split apart into our two daughter cells. So note that in prophase, I'm including prometaphase in prophase right now, but note that our chromosomes are condensed. That's one of the most important parts of prophase. All right, we are now in metaphase, which is one of the easiest ones to identify when actually looking at the cell. So we have our chromosomes that are now going to line up along the metaphase plate, which is just right across the center. So when you see all of them start to move to the center there, you know that we are in metaphase. And that's just in preparation for the sister chromatids to be split apart. The other thing that we're going to see are our centrosomes are now at opposite poles, and our spindle fibers are going to start connecting to the sister chromatids in preparation. The other thing that you're going to notice in metaphase and also in anaphase is going to be these little fibers that are coming out the other side of our centrosome. These fibers are called asters. Right here. And the asters are basically pushing out on the cell membrane in order to create more space for when we move into two cells. All right, another easy stage or phase to identify is anaphase, because our sister chromatids are now separating for the two different cells, the two daughter cells. So remember that with the A, an anaphase is for apart. The two sister chromatids are moving apart. So we'll see them start on the metaphase plate, and the one chromosome that was in an X is now going to be, should I draw it the other way, pulled apart. Now remember that the DNA that is on this side at this point is exactly the same as what is on this side of the cell. However, we're still in one cell. But the main thing is that our sister chromatids have now been split and are pulling apart from each other. Our last phase of mitosis is telophase. In telophase, we're going to start to see the differentiation between the two cells, and we're going to see, so it's really light. Okay, and we'll see our chromosomes starting to unravel just a little bit. And the big thing is that our nuclear membrane and nucleus is starting to reform here, starting to create that appearance of an actual cell again. Finally, after telophase, we move into the last part of the mitotic phase, which is cytokinesis. Cytokinesis is that pure separation of the two cells. It's our final step. So we will see it go from one mother cell into two daughter cells, and our nucleus is now completely formed, and we move back into our normal cell cycle, so back to interphase at this point.